So yeah, welcome to this webinar, co-organized by Tip Biosystems and Lanmer. My name is Dieter Trau. I'm, the founder of Tip Biosystems. Okay, so for today, I want to introduce you into the Photopatter, a handheld spectrophotometer system. And I want to show you some applications. And after this presentation, we can go to a question and answer session. What is a Photopatter? So the Photopatter is a handheld spectrophotometer, and it has a very different workflow and certain unique applications. So the Photopatter is actually a closed system. So it's different to the standard benchtop spectrophotometer that you may have in your lab, where you can use different type of cuvettes from different manufacturers and so on. And the Photopatter is a system built from 3 components: the Photopatter device, then the Photopatter software, which runs on a smart device like a tablet or a smartphone on either Android or iOS, and the Photopatter cuvette tip, which is a disposable that is basically replacing the classical cuvette. So those 3 components, they form a closed system. So what is the principle behind the work of the Photopatter? How does it work? So here on this slide, you see on the left the Photopatter device. And so the Photopatter is a fixed wavelength spectrophotometer. It, in its standard configuration, has 3 different wavelengths. And the light of these 3 different wavelengths is generated by LEDs. And the system is fiber optic based. And the light is guided by a fiber optic through this stainless steel stem to the end of this stem. And also the device contains a light detector, a photodiode, which is collecting light and measures the light intensity. So the Photopatter works with what we call the cuvette tip, which is replacing the classical cuvette. So it's a cuvette, but it's also a tip. So this is how we gave it its name, the fusion word cuvette tip. And on this slide here, on the picture in the middle, you see how the cuvette tip is attached to the end of this stainless steel stem. And on the next slide, you have some idea about these arrows. This is actually the light that comes from the LEDs. It's guided by the light guides, by the glass fibers, and then enters the cavity here of the cuvette tip. And at the end of this cavity here in this region, there is actually a mirror. And this mirror can reflect the light back. And the light is then collected by another fiber and guided through the detector. So if the cuvette tip is inverted into your sample, here in the middle, your liquid, then this sample is entering this cavity and is filling up this cavity. And this is the area, where you actually measure the absorbance or the transmission of your sample. So this means by dipping the tip into the liquid for at least 3 millimeters, the tip is filled with your sample. And in this internal volume here, the light is going back and reflected back and forth, and the absorbance is measured. Okay, so with these features, that the Photopatter is actually inserted into the sample, but not the sample is coming to the Photopatter, there's a very different workflow. So on the top here in this slide, you see the classical workflow we have with benchtop instruments as shown here. We typically have a sample collection somewhere, could be whatever, industrial manufacturing site or environmental sample or in the cell culture lab. We have a transport or move of the sample to the workbench, to the lab bench, and there we have a benchtop spectrophotometer, which we would now, open the spectrophotometers, and we would transfer the sample into a cuvette, either from plastic or glass, place a cuvette into the benchtop instrument, connect it with a computer, usually a laptop, usually a benchtop computer, and program the measurement here, and then we have the result. So that's a classical workflow. You see in the classical workflow, the sample comes to the benchtop instrument, it's inserted into the cuvette, and then it's measured. So the workflow with a Photopatter system is very different. The Photopatter comes to the sample and not vice versa. You see some examples here. The Photopatter can be used directly in the cell culture hood. Here on this left image, measuring directly, for example, a cell density, typically in a cell culture, a vessel like a T75 flask or something like this. Or it can be used here in this image. This is to measure DNA concentration when DNA is prepared for certain other, processes. Or here directly in the brewery, for example, to measure the yeast density in a brewery, a beer or something like this. Or for environmental monitoring, directly perform measurements at the monitoring site, at the lake or river or something like this. So you see, this is a very fundamental change in the workflow, and this change has certain very big advantages. For example, if you have samples that can change over time, for example, the cell culture, the cells still divide, the cell density is changing, or here in the environmental scenario, where you measure, for example, turbidity, but turbidity samples will also change. They might, aggregate and change their turbidity, or the turbidity caused by algae and things like this, and they still grow. So by not measuring at the site, your sample will actually change. You have a very accurate measurement in the lab, but unfortunately, your sample is not longer your old original sample. So those are the advantages that you can capture, with a Photopatter device to measure directly on site. So the Photopatter is a family of products. Currently, we have 5 distinct family members here in this list. It's a Photopatter Bio for measurements for direct protein and DNA measurements at 260 and 280 nanometer. 340 nanometer serves as a background correction, but also can be used for measurement of enzyme kinetics for the NAH redox system. Then we have the Photopatter cell, again here 340, enzyme kinetics, 570, cell viability, and 600 nanometer for cell density. So for cell density, we have built entire device. This is the most simple device we offer. The OD600 only have 600 nanometer. Then the Photopatter turbidity uses 2 wavelengths, which correspond, to certain norms. The ISO used in Europe and the norm used in the US. So what is very unique what we do at Tip Biosystems, we can build any type of custom device with any three wavelengths in the region of 260 nanometer to 1000 nanometer. So we have built many different custom devices for certain applications in particular industries for our customers. So those here's a list of the key features. Let me very quickly go through this. I mentioned already it's a UV and visible fixed wavelength spectrophotometer. It has a LED light source. There's no sample transfer, as you have seen. And if you change the tip, there's no cross-contamination. We have a very large dynamic range, up to absorbance units of 3. We achieve this with the special, algorithms that are used in the instrument. The accuracy is about 0.003 absorbance units. The device is very fast. You can basically do measurements within seconds. Yeah. That's also something very specific here that we have. We have an ambient light compensation. You remember from your spectrophotometer in your lab, you always have to close it before you do the measurement. We can measure in ambient light conditions because we also measure the ambient light. The instrument is powered by a lithium-ion battery, and which you do not need to charge very often. We can do at least 20,000 measurements. We tested this. And after this, we gave up to test. So this is really sufficient. It runs, we have software for iOS and Android. I come back to the software later. It's very easy and user-friendly. So with the data of your absorbance or transmission, we also save the date, the time, and the GPS position. And you can even make a picture or you can write a note. So all this data is exported as a CSV file, and you can send it to your email or you can upload it to a server. Okay, so we have designed the instrument, and in particular, it's stainless steel stem that you see here. So it's from a high-grade steel, rust-free material. And inside there are the optical, the fiber optics, and it's designed in a way that you can perform measurements directly in almost all containers that you find in the lab. For example, in your cell culture hood, directly here in this T75 flask, even if there's only tiny amount of cell culture in there, you can still measure it. And you can also measure in centrifugation tubes, often we call them Falcon tubes, 15 and 50 milliliter. You can measure in the small reaction tubes, 500 microliter. And you can even measure in a 96 well plate if you have at least about 170 microliter of liquid in each well. And still used in, very often in bacterial culture and yeast and so on. You can measure in Erlenmeyer flask. So even you have very little volume in the flask, you can still measure in a 500 milliliter flask. Of course, for bigger flask, you can also measure, but not. But then there must be more volume present. Okay, so we have a number of applications. I don't go to details here. You can find them on our website of Tip Biosystems. We have them available as application notes. I just want to point out here that they are grouped here in different type of industries, life sciences, environmental, the food and beverage industry, and industrial applications. So let me come to some examples, what the Photopatter can do. And here in the next slides, you will see some calibration curves and some comparison to other instruments. Okay, let's start with direct measurements of nucleic acids, either DNA or RNA. which is performed at 260 nanometer. So you can do this directly in the tube or the sample container where you have your DNA. No sample transfers necessary. Also, the sample loss is very, very minimal. I mentioned to you before that the sample enters the cuvette tip, but if you remove the cuvette tip from your sample, that amount of liquid is flowing back and remains into your sample container. So here we see direct measurements of DNA. So that's a DNA calibration curve we did. And we benchmarked or compared this here, with a microvolume spectrophotometer. One example of those microvolume spectrophotometers would be the nanodrop. There are other of those instruments on the market, and the comparison is very good. Let's come to the next example. Also, a direct measurement. So this means there's no reagent or anything necessary here for a protein at 280 nanometer. And you see here a calibration curve for BSA, bovine serum albumin. And so you see a very, very good correlation of the absorbance as a function of the BSA concentration. And again here, we benchmarked against microvolume spectrophotometer. And so the data is similar, within the normal error range of these instruments. So one more direct measurement here would be, cell densities. For example, here a mammalian cell density in the tissue culture lab, and this is done at 600 nanometer. And in this case, we did calibration using a hemocytometer. Remember, a hemocytometer is a device that will take up a certain volume of your cell culture, and it has a grid on it, and then you have to place it under a microscope, and then manually you have to count, different sectors, a certain number of sectors of this grid, and then you have a certain calculation, and you take a certain average, and then with this, after this procedure, which even if you're quite trained, can take 10-20 minutes or so, you can calculate the cell density. So we use, this is a very classical old style method. It's very accurate, but it's very tedious. And we have calibrated the Photopatter system against this method for many different types of mammalian cell lines. And those calibrations are inbuilt now in the device, and which allows the user to do, accurate measurements actually, within seconds. And what you see here is the absorbance as a function of the cell density. And you see this is what you expect at lower absorbance, lower cell density, you have an absolute linear correlation. And later you have a little bit of flattening of this curve. But please recognize here, we already have absorbance values of 3, so which is very, very high. The advantage is that you can, without to dilute your samples with a Photopatter, you can measure very high cell densities. Most instruments, they can measure probably about 2.5. If I say this, this sounds quite a small difference, 2.5 to 3, but actually it's a very big difference, because remember, this is not a linear scale. The absorbance is a logarithmic scale of the light absorbance. Okay, so some other applications, they actually require, certain reagents or so. One would be, to perform enzyme assays using the NADH, NAD redox system. And those assays are very popular in the food and beverage industry, for example, to measure, for example, ethanol or lactate or this type of, you know, small molecular analytes. And here we have a comparison between two instruments, the nanodrop and the Photopatter. And so the assay was performed here at 37 degrees Celsius, but there are different type of after when the assay was read out. So, but both instruments, they perform well. And but here the nanodrop was used in the cuvette mode with 1 milliliter sample, and the Photopatter was used in its normal mode in a microreaction, you know, tube using about 150 microliter sample. Okay, so this is a comparison of different type of samples for this assay. I think I don't need to go into details, but what we can say that the error also achieved with the Photopatter is very low. This is another very common assay that we have in the life science lab to measure protein concentration. It's a BCA assay, which is based on a copper 2+ ion. And this is then forming with protein a colored product, which can be read out as 570 nanometer. That is the wavelengths we have, for example, in the Photopatter cell. And also here you see calibration curve using actually bovine serum albumin, and for a standard, you know, big benchtop spectrophotometer multi-strand from Thermo versus you know, rather small comparable small Photopatter device. So the Photopatter here has a slightly higher slope, which is basically actually an advantage over the other instrument. Okay, so here again, the different sectors where we see, you know, the major applications of the Photopatter. But as I mentioned, we are quite unique company here in the sense we can build a Photopatter custom, and we would be very interested to explore, you know, other applications, you know, outside of these sectors. So we have a couple of application nodes, you know, and growing, available at the Tip Biosystems website in all these industry sectors. We also have some general papers there and some technical notes. So let me come to the software that we have designed, you know, to operate the Photopatter. On the Photopatter itself, there's not much to operate. There's an on-off button, and there's one on top to initiate your measurement. But also you can initiate your measurement in the app. So on this slide on the left, you will see basically the landing page after you open the app. The app is available on the Play Store and online, and there are links on our website. The app can be downloaded by anybody for free. However, you cannot use much of it without the Photopatter device. So if you open the app, you will see this page. And the first thing what you will do is you switch on the Photopatter device and you pair them through Bluetooth. In the pairing process, your smart device will actually recognize which type of Photopatter you have, and then will display the corresponding LEDs or wavelengths of your device, and it will also, very importantly, display all the applications that is supported by your device. Yeah. So this means if you connect different devices, this list here on this image will actually look different depending on the device you connect. So the software is quite, you know, it's very simple. It's very intuitive to use, and as you would do with any other spectrophotometer. The software will guide you and will ask you first to do first to do an auto zero in your buffer or media, and then perform your sample measurements. Then your sample measurements are displayed in this view here. So this is the table view where you see at which wavelengths you have measured, which absorbance and corresponding transmission. However, if you do, continuous measurements, for example, for the enzymatic kinetic application at 340 nanometer, for example, you can also switch to the graphic view by clicking here, this small icon, and then your numbers will be seen in a graphical format. And if you do kinetic measurements, you can, this is very handy and very cool because you can see directly how your results will develop and form your kinetics. So those are the key features of our software. With every measurement, you will also save the GPS position. You can make a picture either from your sample, or if you do environmental measurements from your sampling site, you can write notes, and of course date and time is also embedded. So there's no mix up of those informations because it's seamlessly integrated into the data file. So we have a number of inbuilt applications and calibrations for each of those Photopatter devices. We have another function which is really, really useful. We call this a method maker. With a method maker, the customer can create their own applications or their own calibrations, and they can save them in the smart device actually and can reuse them. So there's a function for automatic measurements to set time intervals. That is very useful for the kinetics. The device is speaking different languages. It speaks English, German, and simplified Chinese. So the data can be very easily exported via email or cloud uploading. It enables you a quasi real-time data sharing with other parties. So let me come to a summary here about the key features of the technology. In terms of portability, so it's ultra portable. It's a handheld device for spectrophotometric analyzers. It makes you independent from a fixed lab position. It's very convenient. It's wireless connected, either through iOS or Android tablets or smartphones. There's no need to transfer your sample. You can directly measure in the sample wherever you are. It's quasi real-time data recording and sharing, and it's super fast. You have your results within seconds. So it has a good flexibility. It supports UV from 260 to almost near infrared to 1000 nanometer. So we can build multi-wavelength devices. Our standard device has 3 different wavelengths. It's very affordable. It's priced only at a fraction of usual benchtop UV spectrophotometers. It's smart. It's basically a IoT, a ready device, and it allows data upload, cloud storage, very, very fast. It's safe, so it's also safe for your sample. There's no cross-contamination between different sample, and we are very eager to explore new applications and opportunities to use the device. So if you order the device, what are you getting? So here you see the different Photopatter devices, the family, and in the picture on the left here, you see the Photopatter kit. What is in the kit is the Photopatter is actually in a nice extra box, which you may keep. The rest of the box you may throw away. And in the box, you find the stand. This is the base of the stand. You find the boxes to keep the kits and to eject the tips from the device. And you find the disposable tips itself. So the disposable tips, also they are supplied extra in a box like this. The tips that you see here, those are our sterile version. They are sterilized with the industry standard process using ethylene oxide. And we also have our standard version, which you see here in this picture. It doesn't mean the standard version it's very dirty. It's not. It's also very, very clean, but we do not absolutely guarantee that they are sterile, but we guarantee they're sterile with the sterile ethylene oxide sterilized tips. So any inquiries, please send them to Lanmer, where you see the email here. And so we are in constant contact with them for their support. So with this, yeah, thank you all for your attendance, and please feel free to ask me any question. Thank you.